Good afternoon, everyone. My name's Alex Cloherty. I'm a Marketing and Communications Manager with Oncode Accelerator. And on behalf of Oncode Accelerator, I'm happy to welcome you to this webinar. Before we jump into the topic at hand, just a few brief housekeeping notes. This webinar will be recorded and transcribed. During the webinar, all of your microphones are muted, but we do welcome questions. So please, at any point during the webinar, you can feel free to type your questions in the Q&A box. And then at the end of the webinar, we have allocated time to address all of your questions and remarks together with our three expert speakers. And before I hand off to the first of our expert speakers today, I'll give a very brief introduction to Oncode Accelerator. We're a program composed of more than 35 public and private partners, and we're funded by the National Growth Fund in the Netherlands. Over the next 10 years, we're working together to achieve our mission, to collaboratively optimize and accelerate the development of new cancer therapies for the benefit of patients of all ages. Our different focus areas in regards to therapy types, cancer therapy types, are small molecules, biologics, therapeutic vaccines, and the focus of our webinar today, cell and gene therapy. So our three speakers today are all part of the cell and gene therapy workstream of Oncode Accelerator. Today, you'll get the chance to hear from Trudy Strijtemans. She's an associate professor at UMC Utrecht and also associated with Darren L. and Rehmed XB. Next, you'll hear from Inge Jedema, professor and head of the translational cellular therapy at the Netherlands Cancer Institute, and also lead of our cell and gene therapy workstream. And then finally, you'll hear from Zsolt Sebastian, also an associate professor at the Lab of Translational Immunology at UMC Utrecht. Now, without further ado, Trudy, feel free to unmute yourself and bring up your slides. We'll be hearing from Trudy, Associate Prof at UMC Utrecht, about how to accelerate the implementation of living drugs into clinical practice. And she'll be focusing in this talk on a GMP simulator facility in an academic setting. Whenever you're ready, Trudy, the floor is yours.
Perfect. Then okay. Then I don't see it anymore. That's the thing. Okay. Okay. Now, thanks for the opportunity to share today with you our approach in Utrecht, how we think to support the acceleration of cell and gene therapy into clinical practice. And I would show you today through the infrastructure we've been building through in the UMC Utrecht. You see my next slide? Just to be sure. Okay, thanks. So in Utrecht, we have set up an infrastructure for advanced therapies. And in that infrastructure, we integrate an innovation, GMP simulation and GMP production. So in the bottom of the slide, you see all the research groups that develop our brilliant new concept that can feed into our innovation or GMP simulation pipelines. And in ICAT, we aim to bring together all the stakeholders in a multidisciplinary infrastructures to develop our new leads further towards patient implementation. And as you are all aware of, these promising new drugs have expanded our medicinal toolbox, but are regulated under the ATMP guidelines. And these living drugs are called, like I said, advanced therapy medicinal products, but they have some features that are different from conventional medicines. They have a more complex and a more diverse diversity. If looking into if you look at the viral vectors or gene delivery technologies that can differ, the formulations being produced in different bioreactors, and most and for all, the autologous nature of many of those medicines bring a variety to these manufacturing. So there's not like basically a one size fits all manufacturing strategy and product development ready. So it will all be risk-based based on the nature of the product. So I would like to take you along in our own example in preparation for first in human clinical trial, which the concept that laid in the Kubel Laboratories group in the UMC Utrecht. And based upon the concept of gamma delta T cells and their receptors that sense metabolic changes in tumor cells, unlike the conventional T cells that recognizes overexpressed or mutated genes in the context of MHC. So the advantage is here not having to deal with a genetic background of the patients, and we can also target different tumor identities. So if you want to know more about this concept, please check out our latest review in Nature Review Cancer of this year. So what we do in the lab is had different tumor-reactive gamma delta T cell clones from which we isolated their T cell receptor sequence. So we brought them in a retroviral vector and engineered conventional alpha beta T cells. And what we see if we introduce these receptors, the endogenous receptors, downregulated, and those cells we call tags. T cells engineered to express a defined gamma delta T cells, a so-called next generation of CAR T cells, whereby we uncouple the gamma delta innate like receptors, and we reprogram both CD4 and 8 cells and use those as very potent killers. And we started a phase 1 trial in AML and multiple myeloma patients by introducing these receptors in the patient's own peripheral blood cells. We purify the cells in our manufacturing process to end up with a pure population to re-infuse back into our patients. So building the clinical study dossier, we think it's very important to have a GMP simulation facility with mainly expertises that are crucial to build all these aspects of your preclinical dossier, such as quality control and analytical assay developers, quality assurance specialists, regulatory affairs, of course, your operators, but also those who know a lot about process development and tissue engineering for those other type of drugs, viral vector specialists and gene editing specialists, all in close collaboration with your GMP production facility to prepare all your dossier to be ready to produce under GMP. And we closely collaborate also in our SIM facility with our national infrastructure there now, in which we harmonize together many procedures, looking into viral vector production and new technologies under GMP, for instance. So we believe that it's very crucial to use a target product profile throughout your development. It is a living document that results in a development plan, a long-term strategy of your own specific product with the end in mind. So how do you want to bring this in the end to the patient? What is your model and how is it scalable, etc. Of course, you need to build your study dossier, your clinical protocol, your GMO license if you go for a genetic engineered product, and you have to produce under your GMP license. Building your common technical document is all composed of these parts. So already a while ago, we published the first concept, we chose our lead TCR, and we worked on the mode of action. But for our true development towards the clinical trial, we optimized our research protocol we added the purification step. We developed models to test our safety and efficacy, both in vitro and in vivo. And these led to our brochure. We believe in the SIM that it's very important to have a structured strategy from the early development phase onwards, from product conceptualization through your validation steps in between your technical runs, your process optimization, have your stability test be performed and leading to a GMP compliant process. We believe also that it's important to follow the quality by design approach. And here's a typical example of a gene engineered process with critical process parameters with your IPCs, your release criteria, all leading to a quality target product profile. So that CMC strategy leads to GMP compliant manufacturing, and we had also to do a molecular characterization of our transgene from the early viral vector packaging cells until the final product that led to our IMPD and the license, our GMO license. And we received regulatory approval to start our clinical trial. And here's a snapshot of the data of our trial. We at the moment in our third dose level, it's not in this slide, but we treated also a first patient in dose level 3 safely. And you can clearly see that we also had some clinical signals, even a complete response in patients, which gives us a lot of hope that this product is not only safe, but might also be very effective as well. So in the meantime, working on the next generation of techs, introducing co-stimulatory receptors and to improve the efficacy. And here you can clearly see in an in vivo experiment compared to our first clinical lead tech 001 that this one greatly reduces the tumor outgrowth in vivo. So what we do in the GMP simulator facility for next generation of tech is amongst others, viral vector development and process validation in the devices we have in our simulator facility. And we are also working on the development and the validation of the new process and their analytics. And with that, I would like to thank our funders, the Kuba Laboratory Group and ICAT that made this possible. And if you have further questions, please reach out to me or ICAT for support and help in your ATMP journeys. Thank you.
Thanks very much, Trudy, for this presentation. And if you have questions, you can also type them already in the Q&A box, and then we'll get to them at the end. We have time for Trudy to answer all your burning questions later on. We'll now start to move on to the next presentation. So Inge, you can start to pull up your slides already and unmute yourself. Next, we'll hear from Inge Jedema, professor and head of translational cellular therapy at the Netherlands Cancer Institute. And she'll be telling us about manufacturing processes for the generation of T-cell therapies targeting different solid tumor types. Take it away, Inge.
Well, thank you very much. I hope you can see my slides properly. Okay, great. Well, thank you for attending this webinar, and I would like to give you some update about manufacturing processes, the generation of manufacturing processes for the generation of T-cell therapies targeting different solid tumor types, and the efforts that are ongoing within our institute in Amsterdam. Doesn't one go to next slides? I don't know why, but my slides aren't moving. Yes, now they are. So if we look at the development of immunotherapy for patients with solid cancer, we can all say that immunotherapies that aimed at boosting the activity of tumor-resident T cells, such as immune checkpoint inhibitions, can lead to robust clinical responses, and they have really been a game changer for patients with solid cancers. However, patients may fail to respond to these kinds of therapy for several reasons. And one of them is that their tumors are poorly infiltrated by T cells. So there are simply not enough T cells in the tumor to combat the tumor. And the other thing is that sometimes there is T cell infiltration in the tumor, but then the majority of these infiltrating T cells appear to be irrelevant bystanders. So they are there, they are recognizing viruses, but they are not recognizing the tumor. And then there's only a very small amount of tumor-reactive T cells. So there is still a big need for alternative immunotherapies that do not depend solely on the quantity or the quality of patients' intratumoral T cells. And for patients with solid cancers, there are several flavors if you look at cellular T cell immunotherapy, and they comprise a method called tumor-infiltrating lymphocyte or TIL therapy, CAR T cells, and TCR T cells. And if we then look at what are the advantages or the features of these kinds of therapy and maybe also the disadvantages, if we start out with TIL therapy, the basis of TIL therapy is that the tumor-reactive T cells are there, but that they are exhausted, and sometimes they are also not there in sufficient numbers. So the basis of the manufacturing process of TIL therapy is that you try to reinvigorate the cells from their exhausted state, and you also try to expand them to make larger amount of tumor-reactive cells that can in the end combat the tumor. And we have demonstrated, and that's published by Marcelo Hahn in the New England Journal of Medicine a couple of years ago, that this strategy can be highly effective in patients with melanoma. And these are patients with metastatic melanoma that have very often already failed first-line immune therapy. And we're now in the process of trying to bring this product towards marketing authorization as NKI. But if you look at the TIL product, it's a bit of a black box because you make an awful lot of cells, and you can think about between 5 × 10⁹ and 2 × 10⁻11 cells, but only a fraction of these cells are truly tumor reactive. However, since we give so many cells, there's probably in a majority of cases there are enough T cells to combat a tumor. However, we don't know for this therapy what is the fine specificity that is being targeted by our product. But we know from research that in multiple cases, very often, multiple antigens are being targeted by these T cells. It's a polyclonal T cell population targeting multiple antigens. And that's more or less the strength of the TIL therapy. If you then look at CAR T cell therapy, this is a bit more difficult for patients with solid cancer. The advantage is that you only introduce one CAR. It's an antibody-like structure that you introduce via genetic gene therapy. And this CAR has a defined tumor specificity because you select it up front. And it's also, this therapy is independent of the quality of the immune cell infiltrate in the tumor itself, since you use T cells derived from peripheral blood, which are healthy T cells in the majority of cases, a bit depending on the pretreatment that the patient has had. But you can make large numbers of these therapeutic T cells. One of the highlights of CAR T cell therapy is that you can only target cell surface antigens that are being recognized by these antibody-like structures. And for solid cancers, it's very difficult to find proper tumor-specific antigens which are not also expressed on healthy cells. And that is what's a bit holding back at the moment the further development of CAR T cell therapies in patients with solid cancers. Although new approaches are out there, for instance, dual CAR T cells and more intelligent way to specifically target the tumor cells. But if we then look at TCR T cell therapy, you have this is also a TCR is also directed against a specific tumor antigen. It's the same with CAR T cells. This therapy is independent of the quality of the tumor infiltrate itself. You can make large numbers of cells, and the advantage compared to CAR T cells is that with TCR T cell therapy, you can target peptides derived from intra as well as extracellular proteins, and you can select upfront your specific TCR of interest with the right affinity. In the NKI, we mainly focus on TIL therapy and TCR T cell therapy at the moment. If we then look a bit further into TIL therapy, what does it entail? So you start with the patient, in this case a patient with metastatic disease that I've tried to show you here. And we start with the surgical resection of one of the tumor lesions. Then it's followed by enzymatic digestion to make a single cell suspension that is put into the initial outgrowth phase under the addition of high dose of IL-2, interleukin-2. Then after this initial outgrowth phase that takes about 2 weeks, the cells are more or less starting to be ready for proliferation, and then we force them into rapid proliferation in the rapid expansion protocol using irradiated PBMCs from up to 15 healthy allogeneic donors, an anti-CD3 stimulatory agonistic antibody, and again high doses of interleukin-2. And this rapid expansion protocol starts in our case in culture flasks and is then followed in the Xuri bioreactor. After 2 weeks of rapid expansion, we have a TIL product that is being pulled in an infusion bag, and that is our drug product. Before infusion of the TIL product, the patient is being prepared for receiving this large amount of cells by lymphodepleting chemotherapy consisting of cyclophosphamide and fludarabine. So that is to prepare the patient for this large amount of cells, to make space and to also make a favorable cytokine milieu for the cells that are being infused. Then we give the TIL drug product, and subsequent IL-2 treatment is given to the patients after the TIL infusion to support the survival as well as the persistence and the proliferation in vivo of these infused cells. If we look at the future perspective, so what are we busy with? What are our ongoing activities in the field of TIL therapy? So we want very much to expand to different indications. We know already that non-small cell lung cancer as well as endometrial cancers will be good candidates, but we will also expand to other tumor indications for the future. Of course, we would like to get rid of these culture plates and these open flasks. So we would like to develop our process into a fully closed manufacturing process. So that's a change that is being prepared within our group. Another thing is that we would hope to get rid of this high dose IL-2 in the culture process by replacing IL-2 in the manufacturing process by other cytokines. Because now with IL-2, you make quite differentiated cells in the end. They're good effector cells, but they are quite differentiated, and they're also addicted to the IL-2, and IL-2 is normally not circulated at sufficient levels in patients. So we think that by replacing IL-2 in the expansion process by other cytokines, we can hopefully also get rid of the in vivo IL-2 treatment that we now have to give to the patients, which is associated with quite some severe toxicity in these patients. Another thing that we would like to, which is an obvious thing that we would like to change, is we would like to find synthetic replacements for irradiated allogenic feeder cells in our process. And of course, we are also working on making better TILs. So making, and we use genetic modification for that. We do this in collaboration also with biotech companies. So we would like to make tumor infiltrating lymphocytes that are able to combat better the hostile tumor microenvironment that may be there in some tumor types, to promote their persistence and to also make TILs with a better effector function. So this is what is ongoing for TILs in our institute. If we then switch gears to TCR therapy, I start off with the viral transduction method. So what we want to do is to bring our transgenic TCR of interest into healthy peripheral blood T cells. And very often we use viral transduction for that. So then you get your expression of your nice transgenic TCR. You can use retro, lenti, or adenoviral vectors for this. And this very often results in random integration into the genome of these T cells. A point to keep into consideration is that there is competition with the endogenous TCR that is also expressed by these T cells, and that will sometimes outcompete your transgenic TCR on the cell surface. And another thing is that for every TCR that you would like to introduce into to make a TCR T cell product, you will need a viral vector and you will need virus, and it's very costly and timely to get GMP grade viral vectors and virus. And to solve that, we are working within DRNL, that was already mentioned, our academic initiative in the Netherlands, to make viral vectors and virus, both retro, lenti, and adenoviral vectors. And these initiatives are ongoing in Groningen, Amsterdam, and Utrecht. And that will really, in the end, speed up and also make it more cost effective to make these T-cell products. Another thing that we would like to point out is that if you only target one antigen, for instance with a CAR T cell or with a TCR T cell, you encounter the problem that there is potential heterogeneous antigen expression within tumor lesions. So not all tumor cells will express all antigens of interest. So we think that it will be better in the future to combine different TCRs in one TCR-T product to avoid the formation of tumor escape variants in patients. But it's not so trivial to bring different TCRs into one product. Because we would like to do that in a way that we don't suffer from the competition for expression with the endogenous TCR that I try to visualize here. And on the other hand, we also don't want to run the risk of mispairing of endogenous and or the transgenic TCR change, leading to potential unknown reactivity, because you then make out of the blue new TCRs that by accident maybe may end up on the cell surface and you don't know what they are recognizing, and that can be very dangerous. So CRISPR-Cas9 non-viral gene editing may be a strategy to solve that. So you start with patient leukapheresis, you activate your T cells, and then via electroporation, you use your CRISPR-Cas9 technology. So we start with using the T cell receptor beta locus specific guide RNAs to make cuts in the beta locus, and then there will be non-homologous end joining, leading to knockout of the beta chain of the endogenous TCR in these T cells. Then we use another guide RNA that is targeting the alpha locus of the T cell receptor that then ruins the alpha locus of the endogenous TCR, and then we leverage this track locus to insert our transgenic TCR of interest, and we do that using double-stranded DNA repair templates and homology-directed repair to introduce get knock-in of our transgenic TCR. So what I visualize here is that we ruin the track locus and then we put in our construct of interest, and that will lead in the end to this edited locus. And in the bottom panel you can see the translated proteins that we leverage, still part of the endogenous TCR alpha locus, but we get a very nice expression of our transgenic beta there. So this is another way of visualizing it. We ruin the endogenous track locus, TCR BC locus, and we introduce our transgenic TCR in the alpha locus. And this is work being done by Brenda Raut and Petra Wawrzyniecka, and this manuscript is currently being submitted. So if you then look how this will work, you again have your peripheral blood T cells. We would like to combine different TCRs in one TCR-T product to avoid the tumor escape variants. So if we apply this technology, what will be the result is that we abrogate the expression of the endogenous TCR, and then we will do subsequent electroporations of different pools. So we will electroporate 1/5 with each TCR if we go for 5 TCRs. And then we use one set of guide RNAs for this, because that's the universal part, and we use different HTR template to introduce our transgenic TCRs of interest. And then in the end, we will have one TCR-T product containing multiple TCRs. And we have now generated a manufacturing process that allows this. And you can, if you have your TCR of wish for your own product, you can come to us and we can turn this into a production process. Since these raw materials for CRISPR-Cas9 gene editing are very expensive at the moment, we also started out setting up a pipeline for academic production of the GMP-grade raw materials. And in the NKI, the focus is at the moment especially at the double-stranded HDR templates, because that is where we really need the speed, the flexibility, and also the cost effectiveness to make new products for every patient. And this is a final slide just to show you that these cells are also functional. So I show you here cytokine production. So these TCR-T cells can efficiently mediate killing as well as cytokine production when recognizing their target antigen. And they can also proliferate and produce cytokines when they are co-cultured with their target antigens. And I will end by acknowledging the team. So this is really a collaboration, like Trudy was also mentioning, between the research part as well as the pharmacy, because you have to close that gap between these two groups to, in the end, speed up the development of new cancer drugs. And that's what we're trying to do. And we also collaborate with biotech companies. I here list two, Miltenyi Biotech and MaxCyte, but there are, of course, more. And we are very open to collaborate with you if you are in the audience. And thank you for your attention.
Thanks, Inge, for sharing all of the expertise that you've built up. I'm sure there will be questions about this. So again, please, to the attendees, feel free to already type them in the Q&A box so you don't forget. Because now we will move on to the last of our presentations. I'd like to invite Zsolt Sebastian to start bringing up your slides and unmuting yourself. We'll hear from Zsolt, who's an associate professor at the UMC Utrecht within the Laboratory of Translational Immunology, about biomimetic strategies for rational choices for immune cell-based drug development. Floor is yours, Zsolt.
Thank you very much. I hope that you can see my slides. And I'm very happy to present here this afternoon. So I would like to show my disclosures. And basically, in my team, we are dealing with the major roadblocks for T-cell therapies, especially in solid tumors. So these are actually choosing the right antigen, providing the best fitness for T-cells, how to deal with proper homing and penetration, and very interestingly, how to deal with immunosuppressive microenvironment. So as Trudy, my colleague during the first talk already introduced, we are very much focusing on the use of gamma delta T cells and especially their receptors. Really a very interesting example that targets very early malignant transformational events in various tissues. So what happens in these transformational events, certain molecules that we called butyrophilins, BTA molecules, they change their surface expression and also their configuration and conformations due to the upregulation of so-called intracell phosphoantigens, and that actually the changes in the surface configuration triggers the recognition of gamma delta T cells. So you can imagine that it is a very complex recognition phenomenon, and as Trudy mentioned, we try to actually simplify it and translate it into a clinical concept that we call tags, when we actually introduce these tumor-specific T cell receptors into alpha-beta T cells. I would like to today bring you through a couple of examples, how we can try to improve the classical original concept by using biomimetic models. So the question is, how we can develop the next generation of tags. As I said, we are very much interested in using the right models for the right question, and that's why, we started from classical 2D tumor cell T cell interactions that are probably best fitting for tumor recognition for screening. Once you introduce, for example, organoids or patient-derived organoids, you introduce an additional complexity to your model. Introducing certain extra matrix components can really help you to identify certain immunosuppressive mechanisms. The same holds true once you introduce additional cell types, for example, stromal cells, that can make your models more and more complex. But that's on its own wouldn't really help you to understand your lead properly. So I would like to introduce so-called multi-modality today that will really help you to enrich your data. So very often we start with the so-called phenome. The phenome is actually the most interesting or most obvious immunological outcome or tumor targeting outcome that you would like to focus on. So think about tumor killing, migration of your immune cells, how your immune cells recognize a target. You would like to improve their exhaustion features, and you would like to make them proliferate more. So obviously, once you have the right phenome, you would like to couple it to a certain signature. So RNA sequencing, a single cell RNA sequencing, can really help you with that. So you can really find the right transcriptomic signature that will correlate to your most wanted phenome. You can even go further. So once you have the ability and you are interested in your composition of your immune cell or T cell subset, you can apply TCR sequencing or even single cell TCR sequencing that can really provide you a so-called TCR-ohm that will give you a certain signature how TCRs contribute to your phenome. And as the last modality, I would like to introduce behavior that I would maybe consider the most complex, where we can really quantify cell-cell interactions, infiltration capacities of your T cells into a certain environment, or various dynamics features. Altogether, or even separately, these features can really help to build your clinical data package. So, for example, you can bring safety efficacy data, you can understand better the mode of action of your lead, you can find new biomarkers, or you can find new leads or co-leads to improve your current therapy. So I would like to bring 3 examples where we try to improve the original tech concept. So how to identify signatures to improve the killing and proliferation capacity, so let's say the phenome of tags, by introducing co-receptors. So to actually increase the fitness of our T cells, so increase the phenome, we tried various co-stimulatory concepts, including ICOS-CD28 or 4-1BB, that we co-introduced next to our gamma delta T cell receptors into T cells to see whether they improve tumor targeting. So what we found indeed that some of these co-stimulatory domains were actually more advantageous than others by inducing interferon gamma production of T cells and also inducing better killing. So we wanted to understand what kind of signatures, transcriptomic signatures, are related to this improved phenome features. So interestingly, once we looked at single-cell RNA sequencing, we understood that there is a rather complex intrinsic heterogeneity that was revealed by single-cell RNA sequencing, which meant that the improved cytotoxic capacity and also the less exhausted phenotype that was a result of these co-stimulatory molecules was mainly affected in CD4 engineered T cells. It was probably due to the fact that CD8 T cells endogenously express the ligand that we targeted here, the ligand for NKG2D. But once we actually targeted BTN3A1 to avoid this competition, as you can see in very aggressive mouse models, we managed to completely cure these mice and had significantly improved survival and long persistence of these T cells. As a next example, I would like to explain to you how we managed to identify signatures, again to improve phenome of our tags by compensating the effects of the tumor microenvironment. So here we actually already increased the system by not only looking, for example, at T cells or tags and the tumor cells, but we also introduced an additional cellular component, stromal cells. So what you can see here, basically we developed a biomimetic system where we actually pre-seeded cancer cells and stromal cells in an extra matrix environment, and through a transfer system we actually applied tags or engineered T cells to this system. What we found is that by changing the stromal composition in this model, we find varying level of T cell targeting against the very same tumor targets. So basically, at the left side of the graph, you can see an reduced tumor targeting, and the more you go to the right, you see stromal compositions that help tumor targeting. So based on that, we defined transcriptomic signatures that will relate to more suppressive stromal composition, as well as transcriptomic signatures that relate to a less suppressive stromal composition. What we found is that these transcriptomic signatures very often lead to changes in extra matrix components. So what we could find is that once we interfere with extra matrix components and T cell binding, as you can see here on this graph, we can actually really radically improve not only T cell migration into the immunosuppressive microenvironment, but also potentially killing of the cancer cells. So this is a good example, you know, how we can use behavior as a kind of final readout in this multimodality. As a next example, I would like to explain to you, how we managed to improve phenome of text by finding tumor-reactive TCRs. So here we made use of various subsets of gamma delta T cells that we found in various tissues in colorectal cancer patients. So here we isolated gamma delta T cells from blood of colorectal cancer patients, from healthy colon tissue, from primary colorectal cancer tissue, or for liver metastasis. So basically, we mainly focused on the so-called B delta 1 population. And what we found indeed is that even though we could identify gamma delta T cells from various tissues, the tumor reactivity of these endogenous T cells, so tissue-present T cells, differed. So basically, infiltrating gamma delta T cells in primary tumor tissue showed the highest tumor targeting capacity. So what did we do? We tried to look at whether we could find any TCR signatures that could relate to this higher recognition. So we certainly found various TCRs that we tested for their reactivity in a TEC format. So we introduced them into alpha T cells. Basically, what we could find is that certain gamma delta T cell receptors that were coming from tumor-reactive gamma delta T cell clones from colorectal cancer infiltrating TILs also kept their tumor targeting capacities once we only use them as a T cell receptor. So basically, this pipeline helped us to find tumor-reactive TCRs. But if we use the very same pipeline and try to look at another model, so looking at transcriptome from the very same different subset of T cells, we found that there are certain features, certain chemokines and chemokine receptors that strongly regulate the presence of infiltrating T cells in certain tissues, especially if it's about tumor-specific T cells. We found that CCR5, a chemokine receptor, is a strong regulator that makes sure that TILs are enriched in colorectal cancer tissues. So using the right model, we could really prove in the lab whether this chemokine receptor and chemokine angle really plays a role in worse or better infiltration by using, for example, patient-derived organoids for such a biomimetic model. And in such a way, we could create a behavior saying that infiltration, for example, from primary colorectal cancer sites towards metastatic site is hampered partially because of the missing chemokine receptor angle. So I hope that I managed to convince you that using not only an increasing complexity-based biomimetic system, but using the right multimodality to combine with, we can basically come up with a plug-and-play design system where we can not only find new targets, but also be able to better identify, the right patients for your treatment, come up with functional signatures that we could use as potential biomarkers. But you could also, use this information to improve your current design to make it better and make it more specific. So with this, I also would like to thank the Cuba laboratories where my team is located and the financial support for this work. And I'm very happy to answer any questions. Thank you very much.
Thanks very much, Zsolt, for this informative presentation. And thanks to all the presenters. We'll bring you all back onto the screen at this point to answer the questions from the audience. So audience members, again, feel free to type your questions in the Q&A box. And we already have one, which is a question for Trudy. So Trudy, the question is, do you work together with the biotech training facility in Leiden for trainings on GMP?
A good question. I think we're open to collaborate with anybody. So if that facility has interesting offers, I mean, please reach out and we can see what we can where we can work on now. Yeah.
Nice. Sounds like a good opportunity to further build up the national infrastructure. Thanks. In the meantime, I had one question for you, Inga. Do you already have some ideas in regards to how you could replace the IL-2 requirement in your manufacturing process, or can you tell us about it?
Yeah, we for sure have ideas. I cannot yet tell very much about it because the manuscript is being in preparation. So we will not disclose it, but there are a couple of usual suspects that are also in the circulation. It's just playing around with the amount and the timing, when to add which cytokine. So it will not be a complete surprise to the audience, but in the end, it's all about the details. And but we're on good on the way.
So stay tuned for a future webinar or presentation to find out more. Got it. Thanks. Zsolt, I also had a question for you. Are there some signatures that you're finding that are highly consistent across different tags or different patients, or is it really patient specific and tag specific that you're the signatures that you're seeing?
Yeah, thank you very much for this question. So even though, you know, in our work, we're primarily focusing on gamma delta TCR and T-cell based signatures, but obviously you would like to make it a lot broader. So many of the signatures that we find are independent of the T-cell lead. So some of them are patient or patient subset specific. But of course, we find also signatures that are very much specific, you know, for a certain lead that you use. So for example, the signatures that I just indicated, you know, in the colorectal cancer patients, that's very much specific for gamma delta T cells. And that's what we also believe, that their gamma delta T cells and their receptors could be a very interesting and promising, you know, approach to treat patients that normally would not respond, for example, on checkpoint therapies, for example, or, you know, classical alpha beta T cell based therapies. So there, I think also the finding those specific immune cell specific signatures is really a very important point. But I think what the Organoid platform and also, you know, this biomimetic platform can provide you is really to look at these signatures in patient specific ways. So you can do that either by classical physical screening or creating new signatures, and also validate them with already existing signatures that are, you know, available out there.
Thanks for the answer, and a good moment for a very brief pitch to the audience that indeed, as well as the cell and gene therapy workstream, we have various platforms within Oncode Accelerator, such as the organoids platform that Zsolt has just mentioned. So we're really trying to bridge these different expertises that are essential for accelerating development of cancer therapies. So for those who would like to learn more, please feel free to visit Oncode Accelerator's website in order to get in touch. In the meantime, while people are thinking about questions, I'll give a couple more moments for people to get over their shyness and ask. But first, I would also like to ask everybody to take a moment to share their feedback about the webinar, where we give webinars on a relatively regular basis with Oncode Accelerator. So we're very pleased to hear what you have to say about this one and how we can improve. As we're waiting to see if any more questions come in, I want to also invite the speakers to, if you would like to give any last words, a couple, anything else that you would like to share to the attendees. Now's your opportunity.
Yeah, well, maybe one small comment is that within Oncode Accelerator, we're very open to collaborate. So we're open to collaborate not only with biotech companies, but also with academic partners. And I think there are quite some opportunities to do so. So I think we have showed just a flavor of what we're doing. But if you have a target or a lead, but you don't have your own GMP facility, I think it will be, yeah, you can be very welcome to reach out to us, and we can always have a discussion about that.
Thanks, Inge. In the meantime, a question also came in for you. One of the attendees is asking to you, Inge, what about the combination of bispecific engagers with TCR therapy? Do you think that's also a good option?
Yeah, I think for the future it will be a good option. But the thing is, you need to know then the toxicity of the two parts of that combination therapy first before you start to combine it. Because otherwise, if you are out of control, you have no clue what is causing it. So I would, at the start, be quite careful with that. But in the end, it will be a good idea. And the same holds true for combining maybe TCR T cell therapy and CAR T cell therapy, or even putting a CAR into a TIL. There are many options that you can think about, but it's always good if you start thinking about combination therapy that you know what you're doing, that you know what your expected toxicity and efficacy profile is, and then start to combine things to improve in the future.
Thanks for answering this. Oh, Zolt, do you also have a comment on this?
Yeah, so if I can just comment on that, obviously it's a very obvious comment that, you know, testing those, you know, toxicity profiles, for example, with for combination therapy. I mean, the models that we developed within Oncode Accelerator, these are actually very suitable models to understand, you know, what your combinational therapies or, you know, leads in certain conditions would really lead to. And I think it's not only, you know, due to the great collection of patient-derived organoids, but also we can make those models the most suitable and most reflective, as Inga said, for the expected toxicity profile. So I thought it was a good bridge to the comment that Inga gave. So once they would be interested in combining bispecific with their TILs, I think I would be very happy to help them to define the right models to, you know, understand toxicity.
Nice to hear. So that's a very nice open invitation to all of the attendees. Then there are no more questions coming in. So if there are no more comments from the presenters, we'll end a little bit early. And I'd like to end with a big thank you to all three of you for giving your time. I know you're all very busy with your research. So thanks for sharing your work with us today. And thanks to all of the attendees as well for your time and hopefully for sharing your feedback. And with that, we'll see you next time. Have a nice evening, all.